It is my great pleasure to introduce our first speaker, who is the founder and the co-organizer of the Fragile Nucleosome, our very own Christine Cucinotta. Christine did her PhD at the University of Pittsburgh with Karen Arndt, where she studied the nucleosome acidic patch and proteins that interact with it. And she is currently a postdoc with Toshi Tsukiyama here at the Hutch, where she is studying how cells wake up from a period of dormancy. Now, in addition to her excellent science that we will hear about today, Christine's passion for increasing inclusivity in science, which is one of the reasons why she started the Fragile Nucleosome community, and her generosity with the time she spends mentoring or even just talking to people in the science community or on Twitter makes Christine a fantastic scientist. I think I speak for all the organizers and the members of the community when I say we are very fortunate to work with Christine and cannot wait to see all the awesome things that she does for the science community. But for now, we will settle for what I'm sure will be a very exciting talk. So take it away, Christine.
Thanks, Roberta. Now I feel a little bit nervous. I feel like the expectations are high now, but I'm just going to tell everyone a cute short story, I think, about how cells can wake up from quiescence. Also, I want to thank the other organizers for doing such an amazing job on the seminar series and just having a lot of fun getting to know everyone and helping, make this area more inclusive. Hopefully, we can continue to strive for excellence and inclusivity. Okay, so as I said, I'm going to tell you a little story about how the chromatin remodeling enzyme RISC in yeast, it's called RISC in mammalian systems, it's known as switch SNF type chromatin remodeler, and how it basically prepares quiescent cells for hypertranscription during quiescence exit. So it's going to be actually like telling the cells what to do to which genes to transcribe on exit. So many cells spend their lives in this G0 state. So normally we have the cell cycle where you think of like G1, S phase, and so on. And then there's actually a stage where the cells can stop dividing and remain either quiescent or senescent or terminally differentiated. And so there are a couple of different flavors of this where we have a reversible G0 where the cells can actually go back and forth between the cell cycle and G0, or we have a type of G0 where the cells remain senescent or terminally differentiated. And this is very important for many different all types of life, ranging from microbes like bacteria, prokaryotes, and then eukaryotes like yeast, for example. And it's very important for surviving starvation, desiccation, contact inhibition, and then in multicellular organisms, one pretty strong example is maintenance of the stem cell niche. So where we have a population of stem cells that remain dormant, and when they need certain signals to either self-renew or divide, then they will exit quiescence and re-enter the cell cycle. Now, this is very important for development, wound healing, immune cell activation, but it's also pretty tricky to get large amounts of quiescent cells and maintain them in quiescence from higher or metazoans, for example. So we actually turn to the budding yeast to try to understand functional mechanisms for quiescence. And so the way we use yeast as a model for this is we can actually grow the cells for 7 days in rich medium, which I will call YPD in this talk. And after 7 days, the cells start to exhaust their sugar levels, and then they either senesce or become non-quiescent cells that are still alive, or they die, or they become quiescent. And the quiescent ones are actually, luckily for us, are quite dense because they have certain storage of sugars and things like that. So we can actually take advantage of this, throw them on a Percol gradient and centrifuge them, and we can actually just like pipette off the non-quiescent fractions and then keep the quiescent fractions. And this is really great for us because we can actually grow large vats of quiescent cells for even like biochemistry, like purify lots of protein complexes from them. So this has been really helpful for us in understanding mechanisms for quiescence and regulation and how chromatin is impacted there. Like I said, we've been looking at quiescent chromatin regulation for a while now, and one of the hallmarks of this chromatin in quiescence is that one, it's transcriptionally inactive. So here I'm just showing Pol II ChIP-seq data clustered by NDR width, and you can see the darker purple color. We have more RNA polymerase bound to these genes. And so what I hope you can appreciate is that there's a lot of Pol II bound in the log cells, which is the actively dividing cells. We call it log for yeast lingo. And interestingly, we also have a broad scale hypoacetylation during quiescence entry. And so what I hope you can see here is when we do H4 ChIP-seq of penta-acetylated H4, you can see that there's a lot less histone acetylation in quiescence compared to log cells. And this was really founded by Jeffrey McKnight, who was a professor at the University of Oregon. And he really spearheaded the work here on quiescence and how chromatin is regulated during quiescence entry. And he is who found that there's broad scale hypoacetylation. The promoters of quiescent genes are narrow, and there's higher histone occupancy and very little to no transcription. And he found that there's actually a master regulator, RPD3, that broadly deacetylates chromatin during entry. My project is I'm trying to understand how cells actually exit quiescence. So how do we go from this broad scale off state to an on state? And so to understand that, when I joined the lab, I decided I would just, purify Q cells and throw them into YPD, or the rich media, and take time points. And I did this, and one of the techniques I looked at was nascent RNA-seq or 4TU-seq, where you can see in quiescence, we have very few transcripts here, which is consistent with what Jeff saw. And then within 5 minutes of adding the cells to fresh medium, we see large amounts of transcription happening throughout the whole genome. And so I hope you can see this on chromosome 5. We have a lot more RNA peaks. Incidentally, we did not see very much, as I alluded to before, we did not see very much Pol II already bound on genes during quiescence, suggesting that Pol II needs to be recruited. It's not really paused, for example, though you do see some straights here. And lo and behold, you can see lots of Pol II binding, which is consistent with the RNA-seq analysis. And I've just done some k-means clustering here to illustrate the changes over time in the different gene sets. And so what I hope you can see here is that there's a lot of transcription going on, and at different types of genes, they get hypertranscribed, and then they kind of go down and dwindle off back to log levels. This, as I alluded to before, quiescence is really important throughout mammalian life, but also hypertranscription is too. And so this is a nice review from Dr. Michelle Perchard, who has participated in our journal clubs, and she will actually also be giving a talk later in our series, but she has a really nice review about hypertranscription. There are just a few highlights here. We have an example where we go from zygotic genome activation and then, like, for example, lymphocyte activation in adult animals. So trying to understand how chromatin is regulated through this process is the goal of my work. And as I mentioned before, we have very low levels of histone acetylation, but what was interesting to us was that we didn't have a huge amount of acetylation occur during quiescence, during the early time points of quiescent exit. So this actually didn't reflect the huge surge of transcription that happened during the early events of exit, suggesting that actually transcription might be occurring despite low levels of histone acetylation. And this is consistent with a recent paper from Ben when he was in Leanne Howe's lab, where he found that transcription is actually what shapes the acetylation across the genome. And so what I hope you can appreciate is that, yes, we do see some little bit, but it doesn't really reflect the same amount as we see in Pol II binding. So how does this happen? What is actually driving this process? And so I wanted to look at chromatin, of course. So as I mentioned before, chromatin is very repressive in quiescence. And we were curious about nucleosomes that might be ready to be removed. So we wanted to look at high versus low digestive MNase-seq data, so we can see, okay, in quiescence, we have this nucleosome here, which is also called, has been called recently fragile nucleosome, and you can see in 10 minutes this goes away. And this is actually quite MNase sensitive, suggesting that this nucleosome might be rapidly removed, where we don't really see a huge difference when we just look at the MNase digestion at the higher levels of digest, suggesting that this might be an important change during quiescence exit. And one of the big factors involved in either binding to fragile nucleosomes, creating them, or removing them is RISC. RISC is a conserved chromatin remodeling enzyme. It's responsible for sliding and ejecting nucleosomes to create NDRs. It's been known as the NDR creator. It's also known in humans as PBAF, and importantly, and we also learned a bit up from Sarah Hayner and Maggie Chasse earlier in the series about this, and it's mutated in all cancers, 20% of all cancers. So that's a really important factor. And I wanted to know, does this actually play a role in regulating quiescence exit? So to do this, I wanted to look at how does it bind in quiescent cells. Now, the cut and run versus chip and RISC aficionados might have some questions about this. Feel free to ask during the coffee chat or in the Q&A. But what I hope you can see is that when we chip RISC, there's a huge difference between log and quiescence and how RISC is localized. RISC really seems to squish into the NDRs in quiescent cells. And this is consistent with a really nice paper that came out a couple years ago from Frank Pugh's lab that saw this similar phenomenon in heat shock, but this was actually quite transient relocalization to NDRs. And what we see here in quiescence is it's very stable. It's actually at almost all of the NDRs throughout the yeast genome. And so I'm able to deplete RISC. So in order to do that, or after doing that, we can see NDRs narrowing. We can see nucleosome positioning in gene bodies weaken during quiescence exit. And then histone occupancy increases in the NDRs as well, which I'm not going to show you for the sake of time. But this raises the question, does RISC promote transcription during exit? Now, RISC has strong roles in transcription during log, and we see this recapitulated here during quiescence exit, where we see, if I'm showing you the RISC in the plus RISC condition, where we have this hypertranscription event, and then again, it diminishes over time. But what I hope you can see is actually at the 10-minute and 30-minute time points with and without RISC, we can actually see not only does it stay low, it doesn't even get to the 10-minute level at 30 minutes compared in the absence of RISC. And and I want to point out that also the shape of the Pol II metaplot is quite different. And I'm going to be addressing that in a minute. So here we're seeing that Pol II is initiation, or Pol II binding is low, suggesting that it has a problem in initiating, which is consistent with a lot of the work that has been shown by David Shore's lab, David Clark's lab, and many others, suggesting that Pol II, and Brad Cairns, of course, Pol II requires RISC for binding. But that's not the end of the story. There's actually quite a bit of other mutation or other defects that we see when we deplete RISC. The other interesting thing was I was curious, like, why is RISC binding to NDRs in Q and then having this effect during exit? And what was interesting is that when I see RISC binding in Q, we have RISC binding at the NDRs, and then during exit, it actually spreads out a bit. It leaves the NDRs, suggesting that it's like actually moving with transcription. So to test this, I blocked transcription by using 110 phenanthroline to shut that down. And then what I hope you can see is that, yes, RISC binding is a bit lower, but then what I hope you also see is that it's kind of getting stuck in the NDR, suggesting that RISC is actually relies on transcription to move out into the gene bodies. So does this actually mean that RISC is acting on gene bodies during exit as well? So to address this, I looked at a subset of genes where I noticed there's this really weird Pol II profile. So here we're looking again at CHIP in 5 minutes. So we have RISC binding, and you can see it's moving out at this region here. And then Pol II kind of getting stuck here at this TSS, and it's still initiating, or it's rather binding. And it's a little bit, there's less in the gene body again in a bit more at the end, which we can talk about at another time. But basically what I hope you can see is that risk seems to be potentially helping Pol II getting past this point here. So it's probably doing something in the gene bodies as well. So not only is risk promoting proper binding, but it's also allowing Pol II to get past this region in the gene. So, but we noticed two other defects in terms of transcription when we depleted risk. So we noticed aberrant transcription happening. So I'm looking at nascent RNA-seq data RNA-seq data where I've taken the log-to ratio of minus and plus risk. So everything in yellow is higher in the absence of risk, or transcripts that are higher in the absence of risk. And I hope you can see at these genes, you can see there's a lot of transcripts happening upstream of the TSS, suggesting that there's actually some aberrant ectopic transcription upstream of the TSS. And this actually falls in line pretty well with how the chromatin is affected. So here I'm just zooming in on one gene. So note the directionality. So here is the transcription start site, and it's going in this direction. And I know this might seem like a lot to look at, but what I hope you can see is, okay, risk is here in Q. We have the risk chip there. And here I'm showing the nucleosomes in Q and in 10 minutes. And what I hope you can see is, and blue is in the presence of risk. So you can see this nice NDR happening here where the gene should be starting, where the promoter is. And then in the absence of risk, we see this filling in as one would expect when you get rid of the NDR creator. And then you see a reduction in transcription here, likely due to the fact that there's very little to no NDR there. But what I hope you can also see is that this NDR starts to form in the absence of risk, and we're actually getting some upstream transcription happening. So this, I believe, is due, and we see this a lot, and I think it's due to the strong transcription event that happens. So when we get rid of risk, not only are we removing the ability to transcribe properly, we're also allowing Pol II to go where it wants to go, basically, or where it can. So we're getting a lot of transcription at the wrong places. And this is also emphasized by the antisense transcription that we see. So there's also quite a few genes, I think it was like about 900 if I remember correctly, where we see an increase in antisense transcription. So here I'm just showing two different clusters. This is kind of like a control cluster down here. And what you can see is that in the sense direction, there's very little transcription happening in the absence of risk, and then we see an increase in antisense transcription. And this falls in line with the narrowing of the NDR, and there's also, as you can see, a 5' push towards the NDR and the nucleosomes and the gene bodies here, preventing sense transcription from happening. And then when there's another class of genes that seems to be a bit more resistant to risk depletion. And these are a lot of the genes that actually have the fragile nucleosomes and can be rescued by, or can be expressed in other means. A lot of these are like divergent genes that have general regulatory, like RAF1 binding sites. And so you can see there's actually a little bit less of a nucleosome problem, and we're not getting as much antisense transcription here. And yes, there's my little cartoon showing that. And then to summarize this, I don't have my clock on here, so I hope I didn't go over. To summarize this seminar, I just wanted to show a quick cartoon about this. So in quiescence, we're seeing risk binding to the genes where there's very little to no transcription. During exit, it starts to spread out into the +1, +2 nucleosomes, which is consistent with what some labs see during log, and then we get nice normal transcription. However, when we get rid of risk, as one would expect, we would block Pol II from binding. Pol II has some struggles getting past the +1 nucleosome, +2 nucleosome, and we also get, basically due to the strong transcriptional response during quiescence exit, we get a lot of aberrant transcription. So either from potential upstream start sites or antisense transcription where it co-ops an available NDR to allow for transcription to happen. And with that, I would like to thank everyone in the Tsukiyama lab, especially Rachel Dell, who's a technician who works with me in the lab, and she's amazing. and former lab member Kian, who initially discovered that risk binds to the NDRs and quiescence. Sam and Jeff McKnight, of course, and then labs that have helped us out with like good science questions and technical help. With that, I would like to thank you all for your attention and thank you for joining us in general in the seminar series too. So love to hear your thoughts on this.
Thank you, Christine. That was wonderful, but we are running a little short in time, so I'll ask a couple
Okay, I'm
questions that
sorry.
we've got. It's okay. I'll ask a couple questions we've got,
Even I can't do it.
but just a reminder that you can stay back for the coffee chat if you'd like to chat more with Christine. All right, so I'll start with a question from Natalia. Great talk. Do you think risk might also help to promote histone acetylation?
I haven't tested that. Maybe.
Okay.
Good
All right, and
question.
then Jason Fan has raised his hand, so you should be able to talk now to ask your question.
Okay, can you hear me?
Yeah.
Yeah. Hi, Christine. Very nice talk. I was wondering, like, just based on the exit from quiescence, I'm guessing like dropping in rich media. Like, do you think like risk has some kind of like maybe nutrient or glucose sensor that responds to it that activates it, or how would that play a role in it kind of like facilitating the exit?
I'm extremely excited about that idea, and I have some thoughts that I haven't tested really. I think it gets modified in some ways, and I'm going to leave it at that.
Thanks.
Thanks for your
Okay.
question.
We'll do one more question, and then just a reminder, everyone can also post their questions on Discord, and Christine can answer your questions there as well if you don't have time to stay for the coffee chat today. Okay, we'll do a question from Constantine Milonas. Great talk. You showed at the beginning that nutrient addition to Q cells causes hypertranscription. However, genes in cluster 2 and 3 display reduction in transcription levels over time. Have you looked at these clusters in your plus over minus risk data?
Wow. Yes, I think so. It's been a while since I've looked at that though, and so there are some genes that go up in the absence of risk. Whether they fall exactly in those clusters, I'd have to go back and look, but it's definitely an interesting question.
Okay, great. All right, so we'll move on to the next speaker.